Now that we've made our messenger RNA, let's go ahead and change that into a protein. In this process, we're going to translate. We are going to go from the language of nucleic acids to the language of amino acids. And we do this by reading in sets of 3 the base pairs in the messenger RNA. And then the cell will read that and interpret that as a particular amino acid. So in translation, we are continuing our process from DNA to the phenotype, the production of proteins. And so up in this diagram, we've added a DNA molecule with a series of genes, and one of those genes is coded in blue there. We have our template strand. Now, I didn't talk about the template or the coding strands during translation, so I'm going to talk about that here. That top one is the template, which means that's the one that's going to be used to make the messenger RNA. On the DNA, the strand that runs 5' to 3' is actually called the coding strand. So that's the actual gene that is being read. And the reason that that one is called the coding strand is because it has essentially the same sequence as your messenger RNA, which is going to be used to make the amino acid. Now, the reason that we use the other one as the template is that we're making the complementary base pair sequence in the messenger RNA. And we want it to be essentially the same as the coding sequence. Now, remember, one of the big differences between DNA and RNA is the presence of uracil. So if you look at that DNA strand, when we transcribed it, we inserted a uracil everywhere there should be a thymine. And so the uracil is complementary to the adenine that was in the original template strand. But in our coding strand, we have uracil. During the process of translation, the messenger RNA is read in sets of 3. So every 3 nucleotides is called a codon, and this reading in sets of 3 is called the triplet code. So every 3 base pairs in your messenger RNA is going to code for one specific amino acid. Now remember that start codon that we had at the start point that we had in our promoter sequence in transcription. That start point is actually what we call a start codon, and it is a set of 3 nucleotides that's going to say this is the beginning of our sequence. And that sequence is important because it sets the reading frame. So now if you think about what happens when you read, if you start at the beginning of the sentence, the beginning of the first letter of the first word of the sentence, and you read one word at a time, it makes sense. But if you shift anything, say your sentence starts with the, and you move over one and you delete the T, and you read in sets, it no longer makes any sense. You've removed a letter from the situation and you shifted your reading frame. So the reading frame is really important because it sets the specific sequence of amino acids for that gene. And if you have the wrong set of amino acids, you're probably going to have a dysfunctional protein, or at least a protein that's not doing what you need it to do in the cell at that time. So here are some examples of what I mean by reading frame. Let's say we have a sentence in triplet code, so these are just 3-letter words. The fat red cat sat. That sentence makes sense, right? All of the words are comprised of all of the letters in the right order, and we start at the beginning with the, we end with our period at the end. But let's say we remove that T, or we start at the wrong place. And if we're still reading in triplet, we now have the sentence hef, adder, edic, ats, at, etc. That doesn't make any sense at all, right? We've shifted our reading frame one, and now if we're talking about proteins and we do this, we might still put in amino acids. We're going to code for something, but it's not going to make any sense in the cell. What happens if we do this in the middle? So if we remove the E in red, the fat, radic, at, it starts out okay, but it doesn't make any sense. And we will talk about how we actually remove or change a letter in the middle a little bit later, but I talk about it here because it illustrates this idea of reading frame. The last example, we can actually change one of those letters. The fat red bat sat. So we've changed cat to bat. Now that sentence still makes sense, and it's actually only slightly different. We've gone from one mammal to the other, but it definitely means something different. So when we change our reading frame, or we change some of the information in that reading frame, we're going to produce a drastically different protein. And so having that start codon at the beginning of your mRNA sequence is going to set that reading frame. It's going to say this is the amino acid that we start on, and this is the triplet code that we continue on. And that's going to give the correct protein for that specific gene. So here we have our genetic code. This is essentially a code table for cracking the triplet reading frame, and we can actually figure out which amino acids are added for each triplet sequence. It's quite easy to use, and we're going to practice using it in class and in lab. But let's first talk about a couple of characteristics of this genetic code. First of all of our sequences start with a start codon. I see there's a plural there. It should not be codons, it should be codon. There is only one, and it is the same no matter what sequence you have. The start codon is always the the nucleotide sequence AUG. And AUG is important because it sets that reading frame. Now, AUG also codes for a specific amino acid called methionine. If you'll notice, methionine is only coded by AUG. There's only one sequence that inserts a methionine. If it's at the beginning, if it's in that promoter region, if it is acting as the start point, the methionine, a very special methionine that has been methylated in a particular way, is going to be put in, and it's going to signal the start of that messenger RNA. If that AUG is not part of that promoter region, if it's not the start codon, then just a regular methionine gets put in there. Now I see a problem that students make quite often, and that is they'll be going along and they'll be making their messenger RNA sequence, and they're going to translate it into a protein, and they start putting in start codons several times in the middle of their gene. Remember, it's only a start codon if it's actually at the beginning. You are not going to see a start amino acid, amino acid, amino acid, another start amino acid, amino acid. Methionine acting as a start codon only happens at the beginning. If it's in the middle of the sequence, it is the amino acid methionine. At the end, we have a series of stop codons. They are highlighted in kind of pinkish orange up there. You'll notice there are 3 of those. The reason that there are 3 of those is that it's really important to stop the amino acid when it's finished. If you continue to make your amino acid chain, you're going to add extra extra amino acids to the end of your chain, and you're going to end up with a dysfunctional protein. So there are several sequences that encode the stop codon to make sure that the sequence actually stops when it's supposed to. And we'll see how this works in a little bit, but it signals the end of the messenger RNA, and so the protein no longer, the machinery in the cell that's adding the amino acids is no longer adding them to the end of the amino acid chain. 2 more important characteristics of the genetic code are something called wobble and something called redundancy. So let's take a look at, we'll start at U, go over to C. If you look here, these 4 nucleotide sequences all code for one amino acid. That amino acid happens to be serine. And if you look, they all start with UC. The third base does something called wobble, and the third base can change without changing the amino acid, or at least it doesn't change it in all cases. This is to allow for some room for mutations to occur without actually affecting the amino acid. So if a mistake happens in the third base of a triplet code of a codon, you can introduce that mutation without even changing an amino acid. So this is really important in continuing to produce functional proteins in a cell. So it actually provides kind of a buffer for mutations to occur, because some mutation can occur without actually changing what you're coding for. Now, not all of them are 4 nucleotide sequences to an amino acid. Some of them only have 2, and that's okay. Most of those ones that are similar codes are actually similar amino acids, and so it still reduces the the problems that you're going to have in a cell, because you might substitute something that's very similar and not really changing the function too much. So this leads us to redundancy. So this is very closely related to wobble, and it's just the fact that more than one sequence can code for the same amino acid. If you'll notice, there are 64 different codes, but there are only 20 amino acids. And again, this just sort of helps with being able to accumulate some mutations that don't do anything, but it also allows for a little bit less specificity, so that transfer RNA that we'll see in a minute that's going to bring in that amino acid can actually do so without being the exact one for the situation. And that, again, that allows for some absorbing of changes, so that all of the mutations that actually do occur don't necessarily affect the coding region of a protein. So let's get into the process of translation a little bit. So remember, we're going to read the genetic code and we're going to build a polypeptide from it. And we do this using transfer RNA as a translator. So remember that I said there are coding RNAs, that's our messenger RNA, and then we have some functional RNAs. These are RNAs that do a specific job in the cell. One of those is the transfer RNA. So the job of the transfer RNA is to bring in the amino acid that is being coded in that triplet code. If we look at the structure here, we have what we call a hairpin structure. So if you think of this as a bobby pin that you put in your hair, they have a little loop at the end and then they fold back on themselves. Each one of these is considered a hairpin. On one end, we have an anticodon. Now, this anticodon is complementary to the messenger RNA sequence that we see that has been made from our DNA. On the other end, we have a different set of triplet. That is the amino acid attachment site. So one end is going to code for a specific amino acid. The other one is going to attach that amino acid to it. And it's called transfer RNA because that RNA is going to pick up an amino acid from the cytosol and actually carry it to the messenger RNA. So the one that is being coded in the next triplet codon is going to come in and it's going to be added to the next chain. This image shows 3 ways that the transfer RNA is drawn. Our book is mostly going to use the right-hand side symbol, which is just sort of a modified cartoon RNA molecule, just because it's easier to see what's going on. So you can see that anticodon at the bottom, that's going to be complementary to our messenger RNA, and then the amino acid attachment site on the other end, which is where the specific amino acid is going to come in to the whole system. So for translation, you need that brand new messenger RNA that you just made from your DNA. You need transfer RNA. Those are all of the transfer RNAs that are attached to different amino acids. So you need a supply of those. And then we need our ribosome. So in chapter 4, we talked about some of the parts of the cell. Ribosomes are present in both eukaryotic and bacterial cells. They're not really organelles. They are proteins that have a function in the cell. Up on the top right, we can sort of see that protein structure. This ribosome is made up of 2 parts, the large subunit and the small subunit. And each of those subunits is made up of proteins and a second type of functional RNA called ribosomal RNA. So this ribosomal, the function of this ribosomal RNA is actually sort of a structural function. It binds with proteins and makes the structure of this ribosome that is actually going to sort of be the workbench for amino acid synthesis. There are two parts. There's that lower part of the molecule called the small subunit. It's down here. And the function of that is, as I said a minute ago, a workbench on which you can build your messenger RNA. So we can sort of think of the messenger RNA as the blueprints or the instructions for the amino acid, and the ribosome is the workbench on which you build it. The large subunit, that top half, is the site where we actually see our transfer RNAs come in, and then the ribosome itself will catalyze the addition of the amino acid to the growing polypeptide chain. And then ultimately you are going to make an amino acid chain, which is then going to be folded into a functional protein. So the ribosome is critically important in both bacteria and eukaryotes because they are the actual molecule that is building the proteins for gene expression. So an additional function is that the ribosome holds the messenger RNA in place so that the tRNA can come into it and sit down on the specific site. Now, ribosomes are what we call highly conserved throughout evolution. What this means is that throughout the course of evolution, ribosomes really have not changed too much. If you have a mutation in a gene for a ribosome, your organism is going to be severely debilitated. The cells are not going to be able to produce proteins. Basically, the organism is not ever going to be able to grow and develop because without the ribosomes, there's no way for the cell to express the DNA. So there are actually multiple copies of the genes that encode the building of ribosomes throughout every genome, and that's because it is so critically important. Ribosome sequences, although they are very conserved throughout evolution, are quite different between bacteria and eukaryotes. And you can actually look at the relationship between organisms based on, or the evolutionary relationship based on the DNA sequence of these ribosome coding genes. Because they are so critically important in the organism, they have remained unchanged over time. And so you can use them for looking at evolutionary relationships, which is pretty interesting. The large subunit of the ribosome has three areas that are important for transfer RNA binding. So we'll see in a minute that the messenger RNA is going to lay on that small subunit, and then the large subunit is going to be sort of a docking site for the transfer RNAs. So we have three sites labeled E, P, and A. That P site is going to be the site where the transfer RNA holding on to the growing amino acid sits. The A site is going to be the site where the next coding amino acid comes in. So transfer RNA is going to come in, bind to the A site, and then it's going to transfer its amino acid to the transfer RNA at the P site. And then actually the whole system shifts. One of those transfer RNAs is now empty and it sits in the E site and then is released from the cell. So you can remember these based on their letters. So the A site is the amino acid site. It's the site where the new amino acid comes in. The P site is the polypeptide site. That is where the chain of amino acids, the polypeptide chain, is going to hang out. And then the E is the exit site. That is where the empty tRNA is going to leave from.